1 1. I think my mic doesn't work. But we'll keep rolling. Day 9. We'll finish this question. I'm still watching this amazing podcast. Really excited about those ideas. One thing I'm excited about is that finally, and thanks to this guy, Michael Levin, we finally can test very interesting theories or propositions or theoretical assumptions, or how I like it to call it, educated guess, that mind can be not only in organical system like humans, animals, and so on, but even in a systems that we never thought before, like algorithms, like idea, a meme. A meme has its own mind to propagate or not procreate, maybe procreate. Very interesting topic, super controversial, which I like, but I really love that you can test it now, and I will be deeper on this more and more. I would love to run some experiments, maybe with software, maybe with other items that might have mind, but I would definitely would love to utilize all my IT experience to run some experiments and be part of this vision, division, vector, aim to actually feel it, or how Michael Levin particularly saying, map out the interface with, or map out a Platonic space. Like I feel like we live in an amazing time. Map out the Platonic space and then have ability to communicate through the thin interface with the, as example, tissues of our body. Like, hey muscles, how about you recover, right? Or hey, you really need to grow one more limb. Very interesting idea. And I really like it that it's not only theoretical anymore, it's actually practical and you can test it, and then based on it there is a open avenue for inventions. Like, you can invent new things every single week. The sky is the limit. Love it. We'll dive deeper into this maybe in next section and next streams and so on. When I comprehend it myself, I'll try to explain. Here's another thing, sir. I tried to explain it to a couple of friends of mine, and I failed miserably. Like, I didn't even get to the point, and the people already get sidetracked to, like, hey, organic is better than chemical. Like, that's not even what I try to say. And I realized that I need so much practice to just simply explain idea or explain what I'm thinking. It's actually very hard. I'm surprised because you expect like everyone be not on the same level as you, but at least, like, you know what's best? I think Richard Feynman said the best. That when someone asked him a question about magnetism, he asked me like, what's your base ground? So he can know what he can use as a foundation to then build up on top of it and explain this or that phenomena. Because when you talk to a person, and if you assume the background is here, right, and start to explain your idea or thought based on this, but the person doesn't know, not doesn't know, doesn't believe, maybe knows better one, that this is a base, so this is already magic. So built on top of it, it's a bullshit. Because whatever you build on something that hangs in the air, nobody trusts that because it's has no base, no foundation. So what you have to do in conversation with person, like, and that's probably a skill, right? That's definitely a skill, and famous people who can communicate well, like Peterson, right? He can probably sense right away the proper foundation of the person where the beliefs are set and then build on top of it. Only that way you can convey your idea. Otherwise, it's all like magic or like bullshit. So that's, I had very interesting experience when I tried to explain his ideas. And I mean, and he claimed very straight that it's not his ideas. Those ideas have been made even before him, but he worked on this and now he finally can practically prove existence of the Platonic space and map it out. He can prove that you're getting more than you put in terms of information. The way how they talk about constant in this universe, that's mind-blowing, right? Like the logarithm, the speed of light, gravity, we have no idea how it is, what it is. We don't even include it into formulas because like it's weak and we literally don't know how it works, why it exists. And his example with the butterflies and Fibonacci numbers or primes, damn, it's good. So I'll be back to his to this particular podcast. I really like his first one, like that happened like 10 years ago with Lex as well, maybe way less, not a 10.5. I remember I was posting to my team saying like, yo, this dude figured out how to regenerate not just tissue, but fingers, limbs, and basically explain that there is a communication of information between cells, if I remember correctly, using electrical signals. I have to rewatch that and to remind myself what was exactly there, but his work is good. His lab, everything like. But back to the bench, right? We do need to finish these questions, know our basis, win the question theory with 52%. Remember, that's not a coin flip. I got upset myself. That's like, hey, I'm kind of answering this question and as good as a random, like the coin flip. You throw a coin and like you're getting 50-50, but I don't have the options, which I particularly chose or set up not to have the options. So I'm not box myself into A, B, C, D. It's a different style of learning. The open book, side tracks. It's like you playing a game and you go into side dungeons and like side missions. It's more fun, I think. More fun, less pressure. And today with such a small attention span in all of us, unless you monk or have iron nerves and stay away from shorts and like from this. Our attention span so limited, so I think that's the only way to study at this point enjoyably, right? Anyway, that's been a long intro. Day 9 is here. Let's let it roll. What is a neuron and how does it send signal? I think neuron is a cell, special type of cell that's first of all provide in organism the way to send information quickly. Neurons can be very long. They send electrical signals. I believe electrical signal between neurons sends as a chemical signal, and they also have action potential. That's what I heard many times. Not super clear what that action potential would mean, but I'm assuming it's exactly what's mean in electricity. Neurons also form neural networks, and it's very similar. I mean, our AI and artificial neural networks, artificial intelligence, it actually mimic the neurons connections in our brain. There is also plasticity of neurons, and I actually don't know, but where we store memory, I think it still debates, is it inside of neurons or not. The thinking itself or reacting on a certain environment triggers or signals like hearing, vision, and so on, that's definitely happening in neurons. But where the storage is, I'm not sure. I get a lot of green boxes. I I hope it's gonna be one. I hope it's gonna be W. Oh no, yellow. Special cell type of sending information quickly, correct. Can be very long, correct. The longest neuron in your body runs from the base of your spine to your big toe. Oh, up to a meter long. Single cell, wow. Electrical signal, correct. The signal travels along the neuron as electricity. Chemical signals between neurons, correct. How to say W right now? Is it West W? I don't know. At the junction between neurons, the synapses, the electrical signals get converted to a chemical signal. Neurotransmitters released across the gap. Call back to question 13. The next neuron receive the chemicals and convert back to electrical. Electrical, chemical, electrical, chemical. Down the chain. Action potential. You said you have heard it, but aren't clear. Here's what it is. Neuron is a rest has a signal. Neuron at rest has a slight negative charge inside relative to outside around minus 70 millivolts. When it's received enough stimulation, ion channels open, positive ions rush in, and the voltage spikes to about 30 millivolt plus 30. This spike is the action potential. It's an all or nothing electrical pulse that travels down the length of the neuron like a wave. Think of it as binary signal, either fires or doesn't, not half fires. Neural networks AI mimics biology, correct historically. McCulloch and Pitts modeled artificial neurons, biological ones, in 40. Whoa, what? And only now we have AI hype? That modern deep learning has diverged significantly from how real neurons work. Yeah, that the whole transformer was revolutionary. Real neurons use timing, have thousands of connections type, and operate on completely different principles than backpropagation. Plasticity, correct. Neuroplasticity is the brain ability to rewrite connection based on experience. Synapses that fires together straighten. Neurons that fires together fire together. Hebb's rule, 1949. Whereas the memory story, you're right that it's complicated, but there is partial answer. Memory isn't stored inside individual neurons like data in the hard drive. It's stored in the pattern and strengths of connections between neurons. So the pattern, this one, according to Michael Levin and what his work is doing, that this actually pattern and strengths, right? It might be an interface from a Platonic space. Think about it. And by thinking about it, you're sending electrical signals through those patterns, and you are part of that thin interface with Platonic space. And who is there? We don't know. But it would be so cool to talk to them. And maybe your thoughts, how you think, maybe that's already how we talk to them. Maybe it's us by driving this body, driving this meat vehicle and trying to figure out what's good, what's not good. Wake up at 8 a.m., going to work, raising children, shaking hands, smile, eat food. All that controls through the interface. Interesting. Very interesting. Very fascinating ideas to think at 10 a.m. Saturday night. Sounds familiar. Yeah. Like I wouldn't trust LLM on this like much. This is way such a new ground, and LLMs are notoriously, I mean not notoriously, that's what the algorithm does. It's a statistical probability. Plus on top of it, bunch of good behavior harnesses. And this is a very important things that people should understand how LLM works, for the reason to know when you can rely on what they're saying and when you can have to actually like hold your horses. And rely on your mind actually. Like writing the class, that's simple. Any LLM can do that. But philosophically think about Platonic space, outsource that to LLM, then might not be a good idea. Sounds familiar, related same concept. I don't think this. What? How come it's like 0.75? I'm gonna ask this because like deeper Weakening one is long-term depression. Your memories are essentially a configuration of connection weights across network. Like, I'm not sure this is true, and they're not sure. Like, what are you like? That was a assumptions, exactly how Big Bang theory was an assumption. It was very good assumptions. Nothing else. What we were explaining how everything begin up until the point we get a Hubble. Oh, James Webb. Sorry, James Webb. And James Webb tell us like, hey, actually, everything what you're thinking here is like not really matching the actual truth. And like, time to invent a new theory, right? So same with this, yes, we have appearance, we have weights, we have, but please cut this out. My throat still with me. I'm gonna like hold on. Why only 0.7? How do you call it? There's all a molecular component specific for so the concept basis never. There's also specific proteins. There is also molecular component ubiquitous region, so it's more likely RNA. Strong conceptual understanding across multiple aspects. You use the electrical, chemical duality, plasticity, and even raise the right open question about memory, missing the mechanism of function potential and synapses details. Core concept may be one important element in one error. Okay, fair enough. Synapses mechanism not described. Neurons structure, dendrites, axons, myelin. I knew about this and this. I didn't know about that. What is CRISPR roughly? Okay, that's interesting. The CRISPR roughly, I don't remember what it stands for, but it's a technology or methodology that's been invented in laboratory. And what it is at some point scientists found out that bacteria attach a small piece of DNA of the virus that attacks the bacteria into their own DNA, into bacteria's DNA through the spacer. So it's basically the small chunk of virus DNA spacer, then another virus DNA, then spacer, another virus DNA spacer. And over time, basically every time when bacteria survive the virus attack, it attaches a small chunk of it to its own DNA through the spacer. And it's exactly the same length. And that was a mechanism for bacteria to kind of build up a memory about different viruses and deal with that better. So scientists found out about it, and they took that mechanism that was in bacteria cells that actually was cutting it by the spacer, was able to cut in this piece of DNA from a regular chain of DNA. And then they hijack that biological mechanism of bacteria to to cut DNA in particular areas. And then they use self-recover mechanism that self-recovers on DNA, and it has two types of recovery. Either just like create a nut and put those, like when you cut out a piece from the long line, it's either try to like put those two loose ends together, or maybe or second part, it's actually put the replacement of the piece that's been cut out. So the scientist and CRISPR's technology itself, it's technology to swap particular chunks of DNA by cutting out the piece and then putting a lot of same small pieces DNA that you want to be replaced with, and utilize cell healing mechanism to take one of those wanted pieces and place it in recovered DNA. Therefore, theoretically, if you can identify in DNA, let's say the color of your eyes, you can change the color of your future baby in embryo by using CRISPR technology. And that can be applied to anything, and most importantly to the diseases that go through the generation, inherited diseases like Parkinson and so on. Maybe not Parkinson, but diseases that comes from DNA, like that pass through generations. The idea is you can use that technology to remove those diseases and basically help out new generation not to inherit something that will hurt them. I think this is one of the longest answers that has been given. We will see. Is this one or is it 0.5? Damn, look at this. This is your best answer of the entire quiz. I don't think it says this if I wouldn't ask him like how do you judge it? Like now it's just pay more attention for like judging mechanism. But again, like it's statistical machine, like doesn't really judge by a significant margin. Like I was getting like 0.9 somewhere. 0.9. Look at this. 0.95 out of 1. Oh wait, that's that's my answer. Okay. No, I did have 0.9. What's this? What was the question? Different. What is the ATP and why does it every biology answer similar? Yeah, see, I had it 0.9 before. One more 0.9. What's the cancer at the cellular level? And this is my what is the ecosystem and what happens if you remove one species from it? This is your best answer yet. I did get 0.9, right? So now I got 0.95 again. And he's saying by significant margin. Like how significant my margin is. Margin 0, 20. This is significant. 40. This is 60. This is significant margin. 0.95 and 0.95, it's not significant margin. Anyway, um, I was trolling. Don't get me. Don't get it wrong. Bacteria store chunks of viral viral DNA with spacers. Correct. The name itself describes this crisp, clustered, regularly interspaced, short, palindromic repeats. I mean, I say this every morning at breakfast to my little kids. Hey kids, good morning. Did you know that clustered regularity interspaced short palindromic repeats was the reason why the regular interspace part is exactly the spacer patterns you described? Bacterial immune memory system. W. Correct. Each spacer is a molecular mugshot of a past invader. Scientists borrow the mechanism. Correct. Jennifer, perhaps to you, Jennifer. Dudna and Emanuela, perhaps to you, Emanuel, figure out how to repurpose it as a programmable editing tool. Nobel Prize 2020. Cast DNA at specific location, correct. The cutting is done by a protein called Cas9. Yeah, I forgot that. Yeah, CRISPR. You guided it to the right spot using piece of RNA that matches the target sequence, like a find and replace where the RNA is the search string and Cas9 is a cursor. Two repairs mechanism correct. Non-homo non-homologous and joining. And cell just glues the loose ends together, often introduce errors which can disable the gene useful for nuts. Homology direction repair cell use provided templates to fill the gap with sequence. This is described. Can change character in the embryo stage. Correct. This actually happened in 2018. So in 2018 it happened, in 2020 Nobel Prize. What happened? Why it's too late? Too late. Why is 2 years delay? Oh, by the way, Chinese scientists, I remember the whole story. I remember the story. It was like more, even deeper experiments, not super legal. And I think like it was some missing people. I don't know. He tried to use CRISPR on human embryos to edit the gene related to HIV resistance. It caused an international scandal, yeah, because the scientist community considered it premature and ethically reckless. Application to inherited diseases correct. Sickle cell disease is the flagship example. CRISPR-based therapy Casgevy was approved in 2023. Oh damn, recent, so recent. 3 years ago. The first criteria for to reach parents. That's how much does it cost? Like I don't know, 70K? What you got wrong essentially nothing. What you got essentially nothing. The only minor Im imprecisions is providing many small pieces of DNA you want to replace it with. You actually provide one template, not many, but the logic of how repair works is correct. How much does it cost? I'm genuinely curious where you learn this podcast, articles, documentary. I mean, I learned this in another tab, right? Because I asked like, hey, what is a what is a CRISP? Yeah, I learned about CRISP in a podcast, but I didn't know exactly the mechanism how it works. And I think in a day 3, I opened a new tab and I learned it. So I remember, and it's definitely not a patterns in my brain. It's something else, or maybe it's patterns, parents. 54. Let's go into three fives. Today we're going for three fives. Let me propagate zone. What are the 4 main types of biomolecular in living things? Oh no, I don't remember that. Oh, you have encountered all 4 during this quiz. Can you name them? I think I can, but how much is it? Oh, this is crazy. Like I have goosebumps. I really thought like 70K is expensive, and it's like 2.2 million per patient. 2.2 million to swap your genes. Hey mom, I think your nose is like too tiny. I want to a little bit like curvy. Can you give me 2.2 million just so I can like, curve it back a little bit? And also roll back a time when I was an embryo. For a single one-time treatment, it's one of the most expensive medicine in the world. Okay, we need to make it cheaper. So not the most expensive gene therapy called Landman for meta-hormonic leukemia cost 4.25 million, and competing sickle cell therapy. Bluebird Lyfgenia is priced at 3.1 million. Whoa. The equality problem is brutal though. Up to 400 new cases of sickle cell appear each year worldwide, most of them in Africa. Where 2.9 million per patient is obviously impossible. In the US, between 50% of sickle cells patients are on Medicaid. So the federal government has been negotiating discount on behalf of state Medicaid. Like what kind of discount you give if a person on Medicaid in the United States? Even if you give 90% discount, a person still have to pay $220,000. And for a person on Medicaid, that amount of money, it's probably 10 years of work. I mean, if it's your life, then yeah. Shaky. Yes, back to the quiz. Back to the quiz. What are the 4 main types of biomolecules in living things? Okay, one of them are lipids, and they're used for membranes, I believe, and something else. Another of them are amino acids. Another one is proteins, or proteins made from amino acids. So yeah, not proteins, amino acids. Then we have enzymes. I think enzymes on maybe enzymes made from proteins. Yeah, maybe enzymes is something higher abstraction. So we amino acids, lipids, nucleic acid, that's what's DNA made from. And then one more, which I don't remember. I have gaps in my knowledge. Biomoleculars, like biology is a really big gap for me. You got two. We're circling a third and missed the fourth. Let me untangle it. Lipids, correct. Membrane, energy storage. Really? Hormones, steroids, nucleic acids, correct. DNA, RNA, amino acids, proteins, enzyme. You keep splitting and recombine this, but they are all one category, proteins. So it is proteins. Proteins are the biomolecule. Okay. Amino acids are the building blocks of proteins. Then, if nucleic acid is the building block of DNA, RNA, then why DNA and RNA not the biomolecule? That's inconsistency between that. You keep splitting and recombine this, but they all one category proteins. Proteins are biomolecule. Amino acids are the building blocks proteins are made from. Enzymes are type of protein. So this whole cluster is one answer, protein. The only you got carbohydrates. Carbohydrates, of course. Carbohydrates, sugars and starches. Glucose, the fuel your mitochondria burn. Cellulose, plant cell walls. Glycogen, energy storage in your liver and muscles. Starch, energy storage in plants. You have mentioned glucose multiple times during this quiz without realizing it belongs to this category. I actually have a side question. Reversing enzyme structure transport, enzyme structure transport signaling. Carbohydrates, sugars, energy fuel, structure, cellulose. Lipids, fats, membranes, some hormones, nucleic acids, DNA. I have actually a side question. So we eat protein, right? Plant protein or meat protein or animal protein or plant protein, say a chunk of meat or fish, or that protein has nothing to do that's with the proteins that our cells use or our body use for everything, like enzymes, structure, transport, and signaling. The proteins that we eating, it's actually, is my understanding correct that proteins that we consume orally, it actually breaking down into smaller pieces in our stomach and then getting sucked in through our digestion system into the bloodstream, circle around all our cells, and then small pieces that's needed, we'll call them, if it's correct or not, I don't know, but we will call them for now nutritions or microelements. Those are getting through the membrane of the cells, and then cells use them as the building blocks to create the proteins that our body needs, using readouts from RNA That's actually a chunk of our DNA. Am I logical chain of thoughts is correct? This excellent, excellent. This is excellent question. Yeah, that's the questions I ask. Every day on this stream, I ask excellent questions. And this is why millions and millions watch. Okay, now being serious. And your logic chain is almost exactly, almost like exactly. It can satisfy without giving compliment. Let me confirm each step. Proteins we eat get broken down in the stomach. Correct enzymes in your stomach and small intestines like pepsin and trypsin chops dietary proteins into individual amino acids. Oh, or small chains of 2-3 amino acids, peptides. Mhm. Absorbs through the digestive system into the bloodstream. Correct. The small intestines lining absorbs the individual amino acids into the blood. Good. Circular to cell. Circulated to cells. Correct. Blood delivers amino acids to every cell in your body. Good. Get through the cell membrane. Correct. Using specific transporter protein. Interesting. So the cell membrane made from lipids, protein channel. That's what I did not know. Cholesterol located in membrane. Peripheral protein membrane. Like some of this, right? Oh. Gets through the cell membrane, correct, using specific protein. Embedded in the membrane, actively transport the cells. Hold on. Gets through the cell membrane, correct. Using specific transporter protein embedded in the membrane, active transport. The cell spends ATP to pull them in. Remember those stored 6 ATPs that's created from every single sugar or glucose? Like, I feel like I show this every single day. That's how we create ATP in mitochondria. Cell use them, and by the way, you know how it calls? You know how, what's the name of that process? I know, just give me a second. Aerobic respiration. Damn it. Anaerobic, that's without oxygen, and aerobic respiration, that's we utilizing O2. Cell use them to building blocks to create new proteins using RNA readouts from DNA. Correct. This is the central dogma completing its full loop in the real-world context. Your cell reads the gene, DNA, mRNA messenger via transcription. The ribosome reads the mRNA translation and assembles the amino acid, which came from the steak you ate, into whatever proteins that gene codes for. So dietary protein and cellular proteins are connected, but not directly. You don't eat a muscle protein and it becomes your muscle protein. You eat the protein, digest it into amino acids. Your cell reassembles those amino acids into completely different proteins according to your own DNA instructions. Your analogy as a programmer. Oh, let's go. Lego. It's like buying a used Lego set at a garage sale. You don't care what the original model was. You break it down into individual bricks and you build your own design from the instruction manual. You denote the bricks as the amino acids, the manual gene. Finish model. Okay, explain me how is this analogy as a program? It's a Lego analogy. There is not even single programmer term here. This is also why complete protein matters in nutrition. Your body needs all 20 amino acids types to build its protein. Animal protein contain all 20. Most plant proteins are missing one or two, which is why vegetarian combined sources rice plus beans together provides all 20. Oh, really? Great synthesis. Think this kind of weight. Does X connect to Y? Question exactly how deep understanding gets built. Question 33. What is the difference between bacterium and fungus? Oh, that's interesting. Let's think together. Bacteria and fungus. Oh, fungus, I think it's a completely different kingdom of our biology or organism. We have plants, we have animals, and we have fungus. And fungus is something between plants and animal. By the way, also separations now starting to hurt us. And this is exactly what Michael Levin highlights, that we should not put a strict border into all our categorization, even between a discipline. It's all a spectrum. He highlights that very beautiful in that Lex Fridman #2 podcast. So, difference between bacterium and fungus. I think the biggest difference is organizational level. Bacterium, even if they live in colonies, they kind of self-minded. Just eat, procreate, divide, eat more, and so on. Fungus, however, I think it's multicellular and it has a, I wouldn't say mind, even so I think it has a mind. Again, Michael Levin like probably would come up with some experiments how to prove it, but maybe it's already proven. What is the difference besides the organization level? Yeah, I can only come up with like the organization, like society level. Bacterium is self-centric, each cell is self-centric, and fungus is society-centric, like the whole organism or the whole colony is the organism. I might be completely off here, most likely. I'll come back to it. Let me score the biology first. Is it biology? Okay. Fungus are separate kingdom, correct. That's the only thing that I get correct. You're right about the major kingdoms. Animals, plants, fungus. Okay. Plus bacteria and archaea, what is, which is their own kingdoms domains. Really. Between plants and animals. This is common intuition, but not quite right. Fungi are actually closer to animals than to plants on the evolutionary tree. This surprised most people. Fungi and animals share a common ancestor that plants don't share. Evidence fungi don't photosynthesize. Their cell walls are made of chitin, same material as insect exoskeletons, not cellulose like plants. And they digest food externally, then absorb it more animal-like metabolism than plants-like. Okay, bacteria are simple, more self-centered, correct in spirit. Bacteria are prokaryotes, and prokaryotes, that's the cells with DNA spread out in a cell without nucleus and without most of the organelles, specifically mitochondria, I believe. Go back to question one. Nucleus, simple architecture. Each cell is complete independent organism. Yeah, I might be wrong about not the embryons, organelles. Fungi are multicellular, mostly choleric. Many fungi are multicellular, mushrooms, molds, but yeast are single cell fungi. Interesting. What's interesting about yeast is that it can last like 400 years. I actually gonna Google it. What is what what is the longest living yeast? That's what I thought. 800 years old. Damn. The oldest revived yeast is what? 5,000 years old? Donna Manier and 12 others. Wow. This is Facebook article. Sourdough for beginners, so I would not super trust this. This is pretty cool. So the kingdom includes both the multicellular one from a network of thread-like structure called Hyphae, Lalale, Hyphae, which wave together into mycelium, the underground network you probably think of. Is it the fungi that's been used to create a Tokyo underground train system? Is it true that in Tokyo to create a underground subway system or metro system they used fungi to design? It's true that in Tokyo they used fungi to Yeah, yes, research uses a brainless organism known as slime mold. I mean, you can tell, right? That's I commuted to work for 20 years, probably 1 hour each direction, and it's a lot of podcasts was going into my ears, like a lot. How do I know this? I don't know. Probably one of the podcasts.
Imagine if a single-celled organism single-handedly designed the Tokyo metro system. Well, this actually happened when a group of Japanese researchers at Hokkaido University put a slime mold on a map of Tokyo. Sounds really strange, I know, but the scientists were actually onto something, and they were astounded by the results.
Well, in Japan.
In Japan, we've been telling about the unprecedented
slime mold
to Japan. But it was only a possibility because
Japanese Tokyo train.
Japan is taking a tougher approach
to Tokyo. Everything in Japan just seems to work. That is the Tokyo metro system, by figuring out the most efficient routes between urban hubs in Tokyo. Okay, I should say that this happened after the metro system was designed, so that it's more like the slime mold replicated it, but don't click away yet, because this is actually even...
So it was actually happened after. But it's interesting that it's replicated. And we're going back to the Michael Levin. That's Platonic space, X-ray, through all the matter, including us, how we would design it, and the slime to achieve the same result. Like, think about it. You don't need to go to school, get PhD, and design a metro system, and same output could be done by a slime. And I'm not saying replace a metro system designers with a slime. No, I'm just saying we can save some compute and get a free one from a Platonic space. Going back to the original question. The key difference you didn't name, cell type. This is the biggest one. Bacteria are prokaryotes, no nucleus, fungus are eukaryotes. Nucleus, mitochondria, organelles, the full package. This is more fundamental difference than colony behavior. A single E cell is more complex internally than any bacterium. Size. Fungal cells are typically 10 times larger than bacterial cells. I had no idea. Cell wall composition. Bacteria use peptidoglycan. Fungi use chitin from insects. This medically antibiotics target peptidoglycan, which is why antibiotics kill bacteria but don't touch fungal infections. You need antifungals for those. Whoa, that's deep. This is so like I knew that you need antibiotics from bacteria and antifungal from a fungal infection, but I had no idea why. I mean, now it all makes sense. It's so clear just to break a cell wall. I mean, hello. Oh, yeah. The 11 point about Bohr. By the way, it's not only the 11 points as well. He emphasized it. But a big fan of Alan Watts, and he was saying exactly the same stuff. Like, hey, we put a net and try to categorize everything. And this is good in the beginning at certain situation, but that's not how this world works. That's not how this universe and simulation works. You know, he's touching on something real. The boundaries between kingdoms are human-imposed categories on continuous tree of life. Slime molds behave like a single cell sometimes and multicellular organisms other times. Lichens are fungus and the alga fused into one organism. Biology constantly resists clean categories. That said, the prokaryotes and eukaryotes distinction is about as hard a line as biology has. It's a fundamental difference in a cellular architecture, not a spectrum. The do fungi have a mind question? This is generally active research. Mycelial network can solve maze. Props to mycelial networks. Allocate resources they can play StarCraft. Allocate resources, build the army, and communicate chemically across meters. Rather than constitute cognition depends entirely on how you define cognition. True. And this is what Michael Levin said as well, which is exactly 11 points. Yeah, I mean, how about to read to the end and then I thought it will be less. What is the difference between passive and active transport across the cell membrane? Ooh, that's good and can be very short. To pass through the cell membrane, you need protein passages. Because from my understanding, only water can pass without proteins. And from passive and active itself, transport across cell membrane, I would say that active, that's the one when cell need to spend energy, ATP, to pull something outside of cell into itself. And the passive, it's when it can go through the protein channel in membrane without use of cell energy, ATP. This is such assumption. I never studied that in biology or in a podcast. Podcast doesn't go that deep. I mean, it depends what the podcast is, but like main one, Joe Rogan and Lex Friedman is like, maybe they are. Nearly textbook perfect. Damn, that was a guess. That was educated guess. 9 days of education and you get an educated guess. Active transport, cell spends ATP to move something. Correct. Passive transport, no energy required. Correct. Protein channels in the membrane. That's what I was showing the images. By the way, I printed it, of course. And do you see these little guys? These guys? That's lipids. Here we go, I will zoom in for you. That's the lipids. Hydrophobic head. I love water. And hydrophilic head. I love water. And hydrophobic tails. I don't like water. Protein channel in the membrane, correct. Water can pass without proteins. Mostly correct. Water can diffuse slowly through the lipid bilayer on its own, but cells also have dedicated water channels called aquaporins to speed it up. So water uses both routes. Interesting. The one detail that completes the picture. Passive transport works because molecules naturally move from high concentration to low concentration down the gradient, like a ball rolling downhill. So it's like the whole entropy, right? And the law of least energy. What's the definition of the entropy and the lowest energy position or potential? How everything fall apart, pretty much. And I don't know why I'm using Google, but entropy is a measure of energy dispersal, okay? Randomness or disorder within a closed system, indicating how much energy is unavailable to do useful work. It is not merely messiness, but rather a statistical measure of the number of possible ways microstates energy can be distributed. High entropy means energy is spread out, which is statistically more likely. Lower energy positions is a configure where the system has the minimum amount of stored energy. Are we saying that water can you want it a high concentration to low concentration? Down the gradient, like a ball rolling downhill. No energy needed because the gradient doesn't work. This is diffusion and osmosis for water. Go back to question 28. Active transport moves molecules against the concentration gradient, low to high, like pushing a ball uphill. That's why it costs ATP. The classic example is the sodium-potassium pump. Every cycle, it shows 3 sodium ions out and 2 potassium ions in, both against the gradients, burning the ATP per cycle. This pump runs constantly in every cell and consume roughly 25 to 40% of your total ATP. Wow, I had no idea about that. I mean, I knew about that process. I knew about sodium and potassium. I did not know about 25 to 40% of your total ATP is what maintains the electrical charge difference across cell membrane, including the minus 70 millivolt resistant potential in neurons that make action potential possible. Go back to question 2. That's why the most energy you spend in your brain, because you need to keep that negative charge inside of your neurons so they can shoot when they shoot or trigger fire. 20-40 of the total TP. Interesting. I want to know like how much of that for brain specifically or for neurons. I almost here. 555. What's it? I'm not gonna read this one. Quick question. I want to know from this 25 and 40 of your total ATP, what is the percentage based on a tissue type? Because I believe neurons would use the most, right? I would say neurons 70, muscles 40, the muscle really wrong. Neurons 60, muscles 30, everything else 10. Think about it. Correct. Neurons are near the top. The percentage varies by tissue because different cell type rely on the pump different degrees, of course. Brain there. Look at called it. Called it. 50 to 60 of the brain total energy. Yes. Constantly fire and need to reset the ion guard gradients after every action potential. The brain is only 2% of your body weight, but it uses 20% of your total energy, and more than half of that goes to the sodium potassium pump. Yes. So this is why your brain is so metabolically expensive. Okay, that's why you need sleep. Kidneys. I mean, first of all, kidneys, it's not a tissue, but it's an organ. And how's this yeah, it doesn't match to 100. Around 60 to 70 of kidney ATP goes to the pump. Kidneys filter your entire blood volume 40 times per day. Whoa! So through the kidney 40 times per day, all my blood, like 4 liters of blood, goes through. And reabsorbing useful molecules, glucose, amino acids, iron from the filtrate back into the blood requires massive active transport. Kidneys are the other organs that disproportionately energy hungry relative to their size. Interesting. Heart muscle, lower percentage go to the pump, specifically maybe 10-15, because most cardiac's ATP goes to the mechanical work of contraction, actin, myosin, sliding. Oh, I saw the animation of that. It's pretty cool. Resting skeletal muscles, relative low pump. So the two organs burning the most ATP, sodium potassium pump, the brain and kidneys. Was essential running ion sorting operation non-stop. Sorting? Where's my skeptical snake? I don't know how to spell snake. Sorting? I need to bring it every time and I call bullshit. I have one more question. Back to question 35. What is the micro? Oh yeah. I have one more question, and that question was about teeth. It's so hard for me to believe. I mean, that's like actually same like bones, but I don't know if it's me for now, but every 7 years, every single cell of our body die and recreated. Is it true for the bones and teeth? Cellular signals as a biological language. Oh, that was interesting research. Let me see. Your hypothesis is not naive. I mean, first of all, on the day 7 or 6, I was proposing that we trying to name every single signal in our body, like the chemical reaction, and we already have millions of them. But maybe it just, since it's a channel of communication, maybe you can say same thing with different chemicals, right? It's like in language, you can say same thing with different words. And what's important for us to learn and understand intent, but not actually words or letters that then build up as sentences and we can comprehend intent. Catokine paradox as the empirical anchor. Like, this is so cool. Like, some person reads this like, oh yeah, this is exactly what it, like, I read this and I understand that in this sentence I know only the, and I know as, and paradox. So much to learn. Oh. I mean, I probably can guess what spherical means. I mean, I probably can understand this better. Anchor, I know the anchor. If I read that in different language. Nevertheless, this is unreadable to me. Oh. Neurotransmission, the closest non-immune analog to language. Yes. Where your hypothesis become most directly defensible is in neuroscience. Thomas Hochfeld established in 1980s that classical neurotransmitters are co-released with neuropeptides. Okay. Function consequences of neuropeptides and small molecule code transmissions. That's what's mind-blowing to me. I'm learning this biology, right? And we always build up innovation by staying in the shoulders of giants, right? Someone was invented something, and then we, based on that, we invented another things, another. Like, that's what means like staying in the shoulders of giants. And all this work that's written, it's probably like dozens of pages. And if I cannot even understand the title, how can I comprehend what they actually saying? I mean, I just hope for AI to basically explain me like I'm 12 years old. And that might work, right? Because the concept is more important, the patterns in thoughts, the idea, but how it's got in your brain through 12 years old basic English, or through this very scientifically friendly language, let's put it this way. It doesn't matter as long as the information passed from one human being through this paper, through AI, to another human being. It's a win-win. Caltech has built the most rigorous experiment framework, Antibell. And this is so recent. Like, look at this. Cell System 2020. Say you'd. Can I visit those labs? Like, I would love to go and visit this lab in Caltech. Oh, that's in like how scientists actually. We're gonna read this. Like, day 10, we're gonna read this. Really quick question. And I probably ask here one time. Okay, really quick question, another sidetrack, but really quick. I always thought that's myth, or maybe not a myth, that's every 7 years your body regenerates every single cell in your body, so you kind of brand new. And I'm okay with like skin, maybe muscles, not sure about brain, that every neuron is regenerated every 7 years. That sounds strange to me. But more concerning is actually like bone cells. First of all, is bones are made of cells, and specifically teeth as well? And if yes, how often they regenerated? By regenerated, I mean like cell die and then another one, or cell divide, some die, and pretty much if we had 10 cells, the process goes on, and then 7 years later, we have exactly 10 cells. Oh my god. But it's a different one. I think I'm done for today. How can it even understood me? Great question, and you are right to be suspicious. The every 7 year things is a simplification that's partly true and partly wrong. The reality is much more interesting. Give me the truth. Different cell types regenerate on completely different schedule. Gut lining replace every 3-5 days. Damn, the fastest turnover in your body. Makes sense. It's constantly exposed to acid, enzyme, and abrasion from food. Skin outer layer replace every 2-4 weeks. Red blood cells, 120 days. I remember that your body makes about 2 million new red blood cells every second. Oh, I did not know about that. 2 million new red blood cells. No way. Is it? Liver cells, roughly 150 to 300 days, but the liver can regenerate massively on demand. Prometheus. Bone cells, yes, bones are made of cells. Wow. Osteoblasts. build new bones. Osteoclasts break down old bone. Your entire skeleton is replaced roughly every 7-10 years. Wow. So the 7 years myth probably originated from bone turnover specifically. Bones are not dead material. They are living tissue with blood supply, nerves, and constant remodeling. Fat cells replace roughly every 8-10 years. Interestingly, the total number stays roughly constant in adulthood. When a fat cells die, a new one replaces it. You don't gain fat by making more cells. Existing cells get bigger. Oh, skeletal muscle cells replace skeletal muscle cells replace roughly every 15 years. Very slow. Now the exception that breaks the myth. Neurons, mostly never replaced. This is a big one. The vast majority of your neurons are the ones you were born with or develop in early childhood, and they stay with you for your life. There is a small amount of neurogenesis in a few specific brain regions. The hippocampus, involved in memory, and the olfactory bulbs, but it's limited and debated. And neurons in your cortex right now is likely the same physical cell from when you were an infant. This is part of why neurodegenerative diseases, Alzheimer's, Parkinson, are so devastating. Lost neurons are essentially gone forever. Heart muscle cells barely replace about 1% per year. Wow. In young adults, declining to 0.3 by old age. By the time you die, roughly 55-60% of your heart muscle cells are original from birth. This is why heart attacks cause permanent damage. Dead heart cells are barely replaced by new ones. Scar tissue fills in instead. Teeth. Here's your most interesting case. Teeth are not cells. Called it. Oh, by the way, all these answers, I'm not sure how true is this. Like this is LLM, like maybe it's a comments from YouTube. Are not cells in the way other tissue are. The outer layer enamel is the hardest substance in your body, and it's almost entirely mineral, calcium phosphate crystals. The cells that made animal amyloblast died after the tooth erupts. So animal is effectively dead mineral. It cannot regenerate at all. That's why cavities don't heal. Underneath the animal, dentin has some limited ability to add new layers from the inside, and the innermost pulp is living tissue with nerves and blood vessels. But the part that matter for daily life is one-time manufacturing job. So it's like coral, right? So enamel is like corals. Why don't we bioengineer amyloblast to regenerate enamel in cavities artificially? Island cells, never replaced. Whoa. Never. The lens of your eye is made of cells laid down in layers straining from the embryo. The cells in the center of your lens right now are among the oldest cells in your body. They gradually lose flexibility, which is why almost everyone needs reading glasses after 45, called presbyopia. So to answer your exact question, every 7 years your brain is grown, your skin, gut, blood, bones, cells fully replaced multiple times over, but your brain, heart, tooth, animal, your framing of 10 cells process goes on 10 cells, but different ones is a class for most tissue. Biological, this homeostatic cell turnover. Homeostatic. The number stays. That's a thermostat operator. Cells okay. The coral analogy is genuinely good. Coral polyps, living cell, secrete a calcium carbonate mineral structure. Then the living part sits on top of the dead mineral it built. Amyloblasts do essentially the same thing, secrete a calcium phosphate mineral matrix, then die once the job is done. Living builders, dead products, same pattern. Why don't we just buy energy back? People are trying, but it's hard for specific reason. People are trying. I'm guessing people trying. Good. The coral analogy is spot on. Coral polyps secrete calcium carbonate and living part sits on top of that. Why it's repeating for the entire living? Why can't we just buy energy? Why it's repeating the same shit? People actually here's what makes it hard. The problem, since animal forming cell are lost after tooth development, alternative cellular sources are required to achieve cellular base enamel regeneration. Just slap the what's the name? Stem cells. Get the stem cells, then tell them that you gonna be animal forming cells. Are you gonna be the cell destruct after building the animal, the program to be disposable. So you can't just turn them back on. You can. Yes, you can. The true challenge lies not in the myosynthesis of the tissue pair, but in the reconstruction of the intricate interface that unifies the tissue into an organ. Oh, the blood and stuff. Yeah. Animals isn't just a mineral coating. It has a precise internal architecture. The hypercrystal, the breakthrough 2025. We closed our guesses almost matching today. Like we so close. Scientists and engineers of Washington and Harvard made strides by using human induced pluripotent stems. Dude, I just said that, like called it. Use the stem cells to regenerate whole teeth. Damn, I'm close. On a day 9, I'm calling last year technology. Think about it. But there was a catch there. IAMs would not mature without help from their neighboring cells. Odontoblasts. Get them too. That's the clever part. The researchers team designed a soluble notch activation called C3DLL4 and computer crafted protein that mimics the natural delta ligand. When added to IM organoids, it successfully matures them into animal secreting amyloblast even without odontoblasting around. So they did it. Damn, good job, guys. So they got stem cells to become in a dish, producing actual animal enamel. But these efforts are still in early experiment stages, with enamel organoids measuring only millimeters. The future vision, researchers are proposing a clinical protocol where the dentist could replace a carious lesson with an precise made of real. No, I don't like this vision. They saying only replace a chunk of teeth with this that's produced from a dish. So instead of replace with the filling, the filling itself kind of comes from organic, not chemical. I disagree with that. Chemical, it's actually probably stronger. I fully want them to regrow the whole tooth. So basically, this tooth get bad, the whole nothing. You take it out, you push stem cells in the proper environment. They do what they do, and like a baby, your new tooth start growing. And then those cells die out, and you're left with mineral or like a coral with a brand new tooth. Probably painful, but that's legit a new set of teeth of yours from your stem cells. Essentially, bioprinting tooth patches. See, like I don't like that patches instead of plastic fillings. But future research is necessary to enhance clinical application and develop. Like it's gonna be so expensive and impractical. So you think, why don't we just ban AMNA is exactly where the field is hidden. We are at the it works in Petrich scale stages. The gap between that is your dentist regrows the animal is probably a decade or more, but fundamental proof good night. See you tomorrow.